CRISPR has dominated gene editing research since Jennifer Doudna and Emmanuel Charpentier's 2012 paper, and could bring huge advances in agriculture and medicine. However, the field has also caused controversy. For example, in December 2019, He Jiankui, a Chinese scientist, got a 3-year prison sentence after news broke of him using CRISPR technology to edit human embryos. But what is the current state of CRISPR now? Where is the progress happening? What are the open problems? And what does its future look like? CRISPR, in its simplest description, is easy to understand. It works by using a guide RNA molecule that targets specific genes and an enzyme called Cas9 that cuts and modifies the DNA at its targeted location. But even more simply, it first finds a specific bit of DNA and then it edits it. According to a report by Global Data, $1.3 billion was raised in 2021 alone from venture capital for gene editing technologies. This marks significant progress, as since 2012 a total of $3.2 billion has been raised, so almost half was raised in 2021 alone, and of the 97 VC deals, 74 of these have involved CRISPR technology. According to the report, most of the funding went to a company called Mammoth Biosciences, which received a total of $240 million. The Innovative Genomics Institute's Fyodor Urnov points to 3 new innovations in the past year that mark significant progress in the field of CRISPR. The first is by the company Intellia, a gene editing company co-founded by Nobel Prize winner Jennifer Doudna, who successfully developed liver-targeting therapies for different diseases by changing only the guide RNA. They changed 20 nucleotides of the guide RNA and got a medicine for a different disease, proving that CRISPR could have therapeutic use in clinics. The second major development is that the two companies, Vertex and CRISPR Therapeutics' collaboration on a CRISPR-based sickle cell disease treatment is expected to be approved this year, likely first in Europe, marking the first approval of a CRISPR-based therapy for human use. Urnov said, "This is not only the year where we've learned that CRISPR is a platform technology, thanks to Intellia, but we're also learning that this platform can be an approved medicine." The third development is what Urnov has called the clinical CRISPR ecosystem, which is the wide variety of CRISPR-based genome editing technologies that are used in research organizations and with patients. Different types of CRISPR protein and epigenetic editors that alter how much protein is made from a given gene rather than changing the DNA sequence are also in development towards clinical use, ultimately increasing the variety of conditions that can be treated using CRISPR technologies. In addition to editing somatic cells, the cells that make up most of the body, it's also possible to edit the genomes of gametes, meaning eggs and sperm, and early embryos. This is commonly known as germline editing. Any such edits in humans would not only affect an individual but also his or her progeny. They could also theoretically be used to enhance desirable traits instead of curing disease. Scientists have therefore called for a moratorium on human germline editing until the serious ethical and societal implications are more fully understood. For example, a 2020 study of genome editing policy in 106 countries showed that 75 of these nations prohibit editing in early-stage embryos for IVF, and not a single country states clearly anywhere that they accept the practice. Sickle cell disease is one of the most common blood disorders affecting millions worldwide. Scientists using gene editing technologies are now able to cure sickle cell anemia. That approach is very expensive and out of reach for the vast majority of the 4 million people who suffer from the disease. So the first real ethical dilemma is that it could in the near future be accessible for people in high-income countries, but much less accessible in low and middle-income countries, which are home to the hundreds of thousands of babies that are born with sickle cell anemia each year. Is our financial inequality going to translate into genetic inequality? In the book The Codebreaker, a biography of Jennifer Doudna by Walter Isaacson, Doudna says, "If you think we face inequalities now, imagine what it would be like if society became genetically tiered along economic lines, and we transcribed our financial inequality into our genetic code." It is a hope of CRISPR researchers that it will benefit agriculture as well, such as the editing of crop genomes, but creating genetically modified organisms also creates the potential for runaway mutations. For example, Gene editing could in theory help to limit the damage caused by invasive species on ecosystems and crop yields, but we are then potentially exposed to risks because of this. As Amy Gutman, PhD, and Jonathan Moreno, PhD, of Pennsylvania University state, if a genetically modified animal or plant escaped, then the gene drive could spread uncontrollably. And if a modified organism mated with a member of another species, it could transmit the changes to new populations. Entire species could be wiped out and ecosystems upended. Another open problem in the world of CRISPR is the unregulated biohacking movement, such as the use of DIY CRISPR kits. Similar to how the early computers in the late 1970s and early 1980s gave rise to computer hacking, the recent accessibility and affordability of gene editing technologies has given rise to genetic biohacking. Due to the fact that most biohackers conduct experiments outside of traditional scientific labs and therefore can't be regulated as tightly, it has the potential to pose serious public threats, such as poor safety, lack of informed consent, and the introduction of unsafe therapies into commerce. Yet, if we asked a biohacker about this, they would say something along these lines.
The stigma of smart people and science is invented. Science works no matter who you are.
If it wasn't clear already, the ethical concerns surrounding CRISPR are likely to accompany the technology for decades to come. But what are the greatest hopes for the future? Researchers have recently used CRISPR-Cas9 technology to perform gene editing in the lungs of mice, which had previously been a challenge due to the difficulty of delivering gene editing tools to cells within the lung. By enhancing lipid nanoparticles to transport the CRISPR-Cas9 system to the tissue, the team of researchers were able to effectively alter the genes for the first time, giving hope in the future for conditions such as cystic fibrosis. Furthermore, a recent breakthrough has showcased the ability to eliminate antimicrobial resistance genes from bacteria using CRISPR technology. This would be huge, as AMR is predicted to cause the death of 10 million people annually by 2050. And most recently, the genome editing mastermind Feng Zhang and his team have developed what some have called the next CRISPR through the use of artificial intelligence and protein engineering. But according to some researchers, the issue that will shape the future of CRISPR most rapidly is the improvement and safety of the delivery, meaning the transport of the segment of DNA into the cells. A common method for transporting CRISPR is through the use of viral vectors. However, these tools in certain instances have caused immune reactions and in some cases transform normal cells into cancerous cells, known as carcinogenesis. The widespread use of lipid nanoparticles shows promise, but we are still far from it being fully embraced. If you enjoyed this video, please subscribe. I also recommend having a look at all the source material I have referenced in the description if you'd like to understand specific research in more depth. Thanks and see you next time.